Hello, class. So we're going to talk about part two of lipid biosynthesis. So in part one, we talked about the building of fatty acids that were saturated fatty acids. And now we're going to continue on with the story of how do our body make unsaturated fatty acids. And so this utilizes enzymes that are in our body where it has three active sites. One is the NADH cytochrome B5 reductase. There you have FAD that is reduced down to FADH using NADH. From there, you have another active site that's called the cytochrome. It has a hemi iron center, and that iron is reduced from iron 3 to iron 2. And then in the final active site, you have the sterol coenzyme desaturase, and that iron that is present is reduced to iron 2. And once it's at iron 2, it reacts with oxygen and therefore produces a double bond with the sterol coenzyme and turn it into oleoyl coenzyme. So it's just the same fatty acid, but just have a double bond that's in the alkyl chain, and that makes it unsaturated. And also the other product is water. So the thing about our enzymes in our body, we cannot produce that double bond no further out than a carbon 9 atom that's in the fatty acid chain. And so how do we get these other fatty acids that that we need that's unsaturated in our body? And so those are called essential fatty acids. And so we obtain that by being supplied in our diet because our body itself cannot synthesize it. And so speaking of a fatty acid that our body does not synthesize is ketone. And so this particular fatty acid is responsible for helping create the lipids that are responsible for our hormones. And that is the icosanoid. With those hormones, you need to utilize them in order to have fever that's in your body, pain, some pain, inflammation, blood clots. All that is part of the icosanoid type of enzymes. So up here, you have leukotrienes, and that is the type of lipid that causes more so inflammation with your lungs, and so causing asthma. So that is also another sort of type of lipid hormones. But utilizing drugs such as aspirin can use it as an inhibitor to not create these lipid hormones that would cause inflammation or fever or blood clotting. And so here are some examples of some derivatives of unsaturated fatty acids that are part of some lipid hormones that causes different changes in our bodies. And all of these are 20 carbon linked fatty acids. So now we have these fatty acids. Now we gotta go more into creating different types of lipids. And so what we have here is just a description of biosynthesis of triacylglycerol. Remember that it has the glycerol as the backbone and 3 fatty acids that are attached to it. And so, as you can read here, most of the fatty acids that are synthesized or ingested by an organism have 1 or 2 fates. One is incorporation into triacylglycerol for storage of energy purposes, or incorporation of creating a phospholipid component of the membrane. And so when your body is in rapid growth, that means it needs more phospholipids that will be made. And so the direction of the synthesis will go more towards making phospholipids. But if your body has plenty of food and the growth stage is more so stunt, then the fatty acids are created with being triacylglycerol, and want to use it as storage for energy for later, since they don't see that these fatty acids are needed for growth or needed to help the body with food supply. So now it moves into a reserve. And with that, with these pathways, when we examine it, you will see that the production of glycerol 3-phosphate is one of the essential intermediate compounds that is required for making both triacylglycerol and phospholipids. And so with this, we'll talk about how do we create triacylglycerol or phospholipid. So at the beginning of stages of creating both is very similar, where you have glucose that you eat in your body and is broken down using glycolysis and produces one intermediate, dihydroxyacetone phosphate. And from there, it goes through a reduction. You see this carbon that's double bonded to an oxygen. It is reduced to a hydroxyl group. And also notice that now the central carbon now is a chiral center. So it has a hydrogen, a carboxylic acid group, a hydroxyl group, and another R group. So each of the groups are different. So you now have a chiral center that is being created with this enzyme activity using the enzyme dehydrogenase. So it produces the glycerol 3-phosphate, and since you have a chiral center, you display what is the actual isomeric structure of it, and that is the L configuration. And there is multiple ways of creating this glycerol 3-phosphate. It can also come from the breakdown of triacylglycerol, where it goes through different steps by, that's called lipolysis, and creates glycerol. And then from there, they have an enzyme activity where it's an ATP dependent type of reaction to make the glycerol 3-phosphate using the enzyme glycerol kinase. And then from there, once you have your glycerol 3-phosphate, now we need to start attaching the fatty acids to these hydroxyl groups, making those ester bonds. So how is that happening? So you have these fatty acids that your body already built up, and you have coenzyme that is available, and so these fatty acids gets attached to the coenzyme, which is an ATP dependent type of reaction, and its enzyme is called synthetase. And from there, once the fatty acid is bound to that coenzyme, it goes through transferase enzyme to start to attach to the hydroxyl group of the glycerol 3-phosphate. And so as you see, there's two open hydroxyl groups, so to go through the process twice to attach different fatty acids. And so from there, it produces a phosphatidic acid. So we're still utilizing this same intermediate compound to make either triacylglycerol or phospholipids. So it's still either or. But now things are starting to break apart. And so you can utilize the phosphatidic acid and the enzyme called Lipid to create a 1,2-diacylglycerol. And just notice that now we have this open hydroxyl group. And remember in the last slide where we had the fatty acid attached to the coenzyme and then go through a transferase to functionalize the hydroxyl group to make that ester. It goes through that same process again. And now that hydroxyl group now has an ester bond, and now we have the final form of the triacyl glycerol. And so as far as the regulation of having triacylglycerol, that is regulated with insulin. And so the insulin kind of promotes having the creation of acetyl coenzyme. And remember what I talked about in the last lecture with the acetyl coenzyme that was in the mitochondria. It got transported into the cytoplasm by the citrates. And then from there, it can actually produce the fatty acids that are being built up in the cytoplasm. So this is stimulated with insulin. But when insulin is not available or not being effective, then instead of creating fatty acid, the body produces ketones. And so that is what is happening to people who have diabetes. And so with this cycle of creating triacylglycerol and having it transported within your body, it doesn't transport as triacylglycerol. It starts off as a fatty acid and would not start off, but it's been broken down into fatty acids. And then from there, it can go into your bloodstream where it can go for fuel for tissues, or it can go into the liver. And once the fatty acid is in the liver, it interacts with glycerol 3-phosphate to recreate this triacylglycerol once more. And then if it's trying to leave the liver and want to have the triacylglycerol go into the fat tissues. So this is what is highlighted in yellow. It's the fat tissues. It also has to be broken down into fatty acid. And so you have the glycerol going into your bloodstream, and you have the fatty acid itself being transported into the fat tissue. And then from there, reacting with glycerol 3-phosphate and recreating the triacylglycerol. So it's always a breakdown of triacylglycerol into either glycerol or fatty acid. Then things can be transported and then resynthesized again. So we just completed the triacylglycerol synthesis and its cycles. Now how do we get to this phospholipid? So still using that same phosphatidic acid and utilizing lipid to have this free hydroxyl group. From there, we need to know that it has that phospholipid phosphoric head that should be attached. And so in order to do that, it uses phosphoric acid and have a dehydration reaction that's happened where it forms the phosphodiester bond, and then there is another hydroxyl group that also goes through dehydration, and that has the head group, and then you have that final form of the glycerol phospholipids, where you have the hydrophilic head and the hydrophobic tail. And with that, that is the conclusion for part 2 of biosynthesis of lipids.